Good morning, students. You all know that the universe is going through a tough situation. The entire planet is under lockdown due to COVID-19 pandemic. In this situation, this is your biology teacher welcomes you for online classes on behalf of Krishnagar Academy. Today, I'll start with the chapter number 3, Absorption by Roots. Before beginning with the chapter, let us have a short introduction on the topic. What does absorption means? Now this absorption means taking in of different substances. In this chapter, first we will learn about the different processes involved in the mechanism of uptake of water along with dissolved minerals. So let us begin our journey of understanding a bit of plant physiology. Now we would like to begin with importance of water in plants. It is an important constituent of the protoplasm. It is also required for several important physiological functions. Now why the absorption of essential components necessary? The absorption is necessary for the normal growth of plants, energy transfer and storage, and for the synthesis of structural components. The minerals that are drawn from the soil helps the plants in many ways. Now, some of the importances are mentioned here. Magnesium, it is an important constituent of chlorophyll. Nitrogen is essential for protein synthesis. Phosphorus is required for growth, nucleic acid synthesis and cell division. Calcium helps in the transportation of substances by maintaining the permeability of the cell membrane. Before going to the subject matter, I'd like to mention that the chapter can be well divided into two parts. The absorption by root part, consisting of the different terminologies, as well as the ascent of sap part with the mechanism of action. So let's begin with the few terminologies mentioned in the chapter. Now the process involved in the absorption of water, if we see it in this way, the transport of water, it is done in two methods. One is passive transport, another one is active transport. Now this passive transport is further can be divided into imbibition, diffusion and osmosis. Now let's begin with what is imbibition. Now this imbibition is the absorption of water by the solid particles of a substance or cell, living or dead, in their dry or semi-dry state without forming a solution. The liquid which is imbibed is called imbibate, and the solid substances which take part in imbibition are called the imbibants. The imbibition increases the volume of the imbibant, which develops a pressure called the imbibition pressure. So here the point which is to be noted is that in this process the particles or the substance, they absorb water in their dry or semi-dry state and without forming any solution. And the next process or some common examples, let's see. Now if you soak some dry seeds, it will swell up the next day. Now this process is due to, or this occurrence is due to the process of imbibition. As because when we were soaking, the seeds were dry, but after soaking throughout the night or throughout the day, the seeds will swell up and the seed coat may burst also. So we can say that it also helps in germination. Now the next process is diffusion. It is the free movement of molecules of various substances, that is solid, liquid or gas, from the region of their higher concentration to the region of their lower concentration when the two are in direct contact. So for diffusion to occur, the substances, the solutions or whatever it is, they should be in direct contact with each other. Some common examples are like when sugar is added in water, its molecules diffuse throughout the liquid, giving it an uniformly sweet taste. Or else if we check like that, the smoke that comes out of an incense stick or the deo that we spread in our room or the room freshener, after some times it's get equally distributed throughout the room. Now the diffusion process continues until a state of equilibrium is reached. Diffusion pressure is the process exerted by the diffusing particles and is directly proportional to their concentration. The significances of this diffusion can be seen in the processes like respiration and photosynthesis, where the gases gets diffused throughout the system, whether it's a plant or it's an animal. In case of respiration, we can see the diffusion of carbon dioxide and oxygen. And in case of photosynthesis, the plants, until and unless they take the carbon dioxide, they cannot produce the food. The next process is osmosis. It is defined as the movement of water molecules from the region of their higher concentration to the region of their lower concentration when separated by a semi-permeable membrane. Now for the definition of this process, we should keep some points in our mind. The first thing is, in case of osmosis, only the water molecules move from the region of their higher concentration to the region of their lower concentration. So in other words, we can also say that it means the solvent molecules move from dilute solution to a concentrated solution. And in these cases, the solutions must be separated by a semi-permeable membrane. Now while talking about these membranes or about the permeability, we can say that these membranes can be of some types. Like semi-permeable is mentioned here. It means it allows only some certain substances to pass through it. The membrane can be fully permeable or the membranes can be impermeable also. So totally permeable or fully permeable means it allows everything to pass through it. We can see such thing, or such an example is the cell wall, whereas semi-permeable membrane is the cell membrane and impermeable membranes are those membranes which don't allow anything to pass through it. Then types of osmosis, it is mainly of two types, endosmosis and exosmosis. Endosmosis is the osmotic inflow of water into a cell when placed in water. It causes swelling of the cell. So in this case, we can see the example like the raisins. When placed in normal water, after some time it swells up. Hence, inside the raisins, the concentration of water was low. So from outside medium, the water has entered inside and has made the raisins to swell up. In case of exosmosis, it is the osmotic outflow of water from a cell when placed in a solution having lower concentration of water as compared to the cell sap, which causes the cell shrinkage. Here we see we have taken some grapes and we have put it in salt solution. So here the concentration of the solution was more, means water was less. So water from the grapes has passed outside and has made the grapes to shrink and it has become shrunken grapes. So the process here is exosmosis. Then comes osmotic pressure. The minimum pressure that must be applied to prevent the passage of pure solvent into the solution through semi-permeable membrane. Thus, greater the solute concentration, greater will be the osmotic pressure, and lesser the solute concentration, less is the osmotic pressure and more is the osmotic movement. Then comes the tonicity. Now, it is the amount of tension developed in a system on account of occurrence of solute particles in it. So, when we can consider about tonicity, when the liquid is a solution and some amount of solute is present there. So, based on tonicity, solution is of three types. The first one is isotonic solution. It is the relative concentration of water molecules and the solute on either side of the cell membrane that remains same. Now, what I said is that here a tension is developed in a system, so there should be some separations or two mediums should be there. So when the concentration of both of them are same, we can call it as isotonic, as iso means the same. Then is hypotonic solution. Hypo means less. So the solution outside the cell having lower concentration of solutes, hence endosmosis occurs. Whereas in case of hypertonic, hyper means more. So solution having higher concentration, solute molecules outside the cell, hence exosmosis occurs. Now the significance of these can be seen in absorption of water by roots and stomatal movements. Now in case of absorption of water by roots, due to osmosis, the water passes from cell to cell. In case of stomatal movements also, we will see that since the guard cells are also cells, water enters inside or water leaves the guard cells to make it turgid or flaccid, which makes the stomata move or the opening or and closing of stomata possible. So next we will read turgidity and flaccidity. Now the state of equilibrium when no more water can enter into a cell is called turgidity, and the cell is said to be turgid, that is fully charged with water. The pressure exerted by the cell contents against the cell wall in turgid condition is called the Turgor pressure. Now, we should keep these terminologies intact. Like that, the turgidity is when no more water can enter into a cell. It is means it's a fully charged with water. So this is, or rather this explains turgidity. So the pressure that is exerted by the cell contents on the cell wall is known as Turgor pressure, whereas the opposite pressure, that is the pressure exerted by the cell wall against the cell contents under fully turgid condition is called the wall pressure. Now, this Turgor pressure and wall pressure in a cell normally counterbalance each other and the state of equilibrium is maintained. The opposite of turgid is flaccid. Depending on turgid and flaccid conditions, the opening and closing of stomata are controlled by guard cells. They are hence often called Turgor operated valves. Then root pressure. It is the pressure developed in the roots due to inward movement of water, which helps in pushing the sap of plant upwards. Due to excessive root pressure, excessive water is lost along the leaf margins called guttation. Plasmolysis and deplasmolysis. The shrinkage of the cytoplasm of plant cell from its cell wall under the influence of hypertonic solution is called plasmolysis. The cell in this state is said to be limp or flaccid. This condition is called flaccidity, and which is the reverse of turgidity. Now, here we can see it was a normal epidermal cell. When it is put in sugar solution, you see the sugar solution has taken the place and the cytoplasm or the cell protoplasm has shrinked, and hence it has become a plasmolysed cell. Again, when the cell is put in water or any hypotonic solution, it regains back its original position, and hence it is known as deplasmolysed cell. So, the regaining of turgidity and normal condition by a plasmolysed cell when placed in a hypotonic solution is called deplasmolysis. Then we will study about active transport and passive transport. Now, active transport is the movement of molecules from its lower to higher concentration through a living cell membrane against the concentration gradient, which requires energy. So, while studying active transport, we should keep in our mind that it's against the concentration gradient, whereas passive transport refers to the movement of molecule requiring no energy from the cell. There is a free movement of molecules from their higher concentration to the lower concentration, as we have seen in diffusion. Then comes the process of ascent of sap with the end of the terminologies. Now, after understanding the different terminologies, we will understand this thing better. The upward movement of water along with the dissolved minerals from the roots to the tips of the stem, branches and their leaves is called the ascent of sap. Ascent means the upward movement and sap is the cell sap. Now, the path which follows is soil water, which enters the root hairs. From there it goes to the epidermis, then cortex, endodermis, pericycle and xylem vessel. We can see in this diagram, these are the soil particles. Here water was available. From there the water has entered the root hairs. From here you see it has passed the epidermis, the cortical cells, endodermis, pericycle, and ultimately it has reached the xylem. Through the xylem column the water rises up and it goes out through the leaves by the process of transpiration. Now, to understand the process of ascent of sap, let's read in details. So, the upward movement of sap is brought about by virtue of four main forces. These are, number one is root pressure, which builds up sufficient force to push the sap in xylem vessel up to a certain height. The next is the adhesion of water, which causes water to stick to the surface of the cells, thus drawing more water molecules from below when leaf cells lose water during transpiration. Next is capillary action. When the narrow diameter of the xylem causes water from lower level to rise in order to fill the vacuum created by the loss of water due to transpiration, narrow diameter of the tube causes capillary force which makes water to rise up. Then comes the transpiration pull. More water molecules gets pulled up due to the tendency of water molecules to remain joined, cohesion during transpiration. A continuous column of water through the stem is maintained. So, by these four processes, actually the ascent of sap process is made possible. Now, one by one, if we see water in the soil, it is taken up by the root hair, it goes to the root cortex, then xylem vessels, upward movement through the xylem in the stem, it goes to the leaves, from the leaves through diffusion, it goes out through the stomata as water vapor, which is transpiration, into the atmosphere. So, this transpiration here creates a pull which due to its suction pressure takes the water up the capillary tube or the xylem, and the rest of the water is pushed here by means of root pressure. So, this was the whole process of ascent of sap in plants. That's all. Thank you.